This is Module 3 for our NAS2 review, and in this module we're going to cover cells. Three main components for cells: cell membrane, and we'll talk about the phospholipids that make up the cell membrane. There's the cytoplasm. The cytoplasm is the cytosol, which is a fluid, and the abbreviation here, intracellular fluid, ICF. So it's the fluid plus the various organelles. Most cells are going to have a nucleus, but there are some cells, for instance, the red blood cell, that does not have a nucleus. Now, the cell membrane is pretty important because it's going to control what comes into and leaves the cell. And I put down here abbreviations to be familiar with. ICF is intracellular fluid, fluid within the cell. ECF is the extracellular fluid, or fluid that is outside the cell. Here we have the cell membrane, and as we looked at in the first module, it is a phospholipid bilayer. Here's a reminder of what a phospholipid is. It has a part of it that is what we call polar, meaning it's going to interact with water. It's going to form hydrogen bonds with water. Then it has a part that is nonpolar, and that is what we call the hydrophobic tail of it, made up of mainly carbon and hydrogen. And it will arrange itself, when you put a bunch of these phospholipids together, it will arrange itself with the hydrophobic part on the inside and the hydrophilic part facing the outside of the cell and the inside of the cell. It'll look something like this. So here on the right is a illustration of a cell membrane, and you'll see some different things stuck in the cell membrane. The blue things here are proteins. So proteins are very important because they can form channels and pumps that will allow different things to go through the membrane. Also, you'll see little peripheral membrane proteins. Some of these are enzymes. We talked about enzymes last time. They make reactions go more quickly. Other things you'll see, things sticking out here, this is a glycoprotein. So if you look at the word, it's got a protein in it, but it also has this glyco. Glyco means carbohydrate. So the carbohydrate here is sticking out, and here's the protein in the membrane. You also have glycolipids. So a glycolipid, the lipid is in the membrane, and the carbohydrate part is sticking out. Another thing you see here, this is a depiction of cholesterol. So cholesterol is a type of lipid, and cholesterol adds some stability to the membrane. Membrane transport. As I said before, the membrane is important in allowing certain things to go into the cell and keeping certain things out of the cell. So what you see within a cell and the composition of that intracellular fluid is going to be different in terms of the ions that you see, the different proteins that you see, than what is outside the cell. How do things get in and out? Well, they can do that a few different ways. If something is small and nonpolar, well, they get through that lipid portion pretty easily, and those are things like oxygen and carbon dioxide. They will go from an area of high concentration to low concentration. We call that simple diffusion. They don't need anything to cross, they just cross it very easily from high concentration to low concentration. Other things that are larger, like proteins and most amino acids, or they are polar, things like glucose. Remember I told you carbohydrates are polar. Well, they can't really get through that lipid part very well because they can't dissolve in the liquid. They're water soluble. So they need to have some sort of channel, a protein channel, in order to cross. The other thing that needs a protein channel are ions. Ions, remember, have a positive or a negative charge to them. They can't move through that lipid layer because they are not soluble in a lipid. They are soluble in water. When something needs a protein channel, but it's still going from a high concentration to a low concentration, we call that facilitated diffusion. Both of these are what we call passive transport, meaning no energy required. Again, passive transport, no energy required, meaning no ATP. Remember, ATP is the form of energy our cells need. Substances are just moving down a concentration gradient. Remember, when you see that word gradient, it just means difference. On the left-hand side here, we have simple diffusion. This is a molecule that can easily cross the cell membrane, and it just goes from area of high concentration to low concentration, and it will keep moving until the concentration on each side is the same. On the right-hand side here, we have facilitated diffusion. Things are still moving down their concentration gradient, but in this example, these substances can't cross the membrane on their own. They have to have some sort of channel. And here's a protein channel that's going to allow whatever this is to cross the membrane, and it will go down its concentration gradient until the two concentrations are equal. Osmosis, also passive, doesn't require energy. This is specifically the movement of water. Water moves from an area of lower solute concentration to higher solute concentration. So here we have two beakers. In the middle of each beaker is, we see a semi-permeable membrane. We have water and we have some solute. Remember, the solute is what gets dissolved in the solvent. So on this side, this is pretty dilute, low solute concentration. On the other side, high solute concentration. Well, which way will the water move? It's going to move from left to right. And it's going to move until those solute concentrations are the same. Movement of water is called osmosis. It's passive. No energy is required. And water moves from low solute concentration to high solute concentration. And this brings us to the concept of tonicity. And we talked about osmolarity, and tonicity is similar because tonicity compares one solution to another. And a lot of times we're going to be comparing extracellular fluid to intracellular fluid. Now, a solution may be hypertonic, which means it has lots of solute in it. It could be isotonic, which means that the amount of solute is the same when you're comparing two solutions. Or it could be hypotonic, where the solution has less solute than one you're comparing it to. And the water will move between solutions. Water moves to a higher solute concentration. The best way to illustrate this is with red blood cells. I'm going to do the middle one first. The middle one is isotonic. Iso means same. This means that the fluid inside the red blood cell is the same concentration as the fluid outside the red blood cell. That means that there may be a little water going in, there may be a little water coming out, but there's no net movement. And if you look at these red blood cells, they don't change in shape or size any. No net movement of water. Let's go over here to the hypertonic solution. And the hypertonic solution is referring to the solution you're putting those cells into. In the hypertonic solution, that solution has more solute in it than what is inside the red blood cell. Therefore, water moves out of the cell into the solution. And these red blood cells will shrink or they will crenate. C-R-E-N-A-T-E. That's the word that's used. So cells will shrink when they're put into a hypertonic solution. Hypotonic. And remember, this is just comparing the solute concentration between solutions. In a hypotonic solution, that solute concentration is less outside of the cell than what's inside. So in this situation, water is going to move into the cell. And eventually, as you see depicted here, these cells are going to burst or lyse, L-Y-S-E. So tonicity just refers to the amount of solute within a solution, and it's comparing one solution to the other. We talked about passive transport, simple diffusion, facilitated diffusion, and then we talked about osmosis, which is specific for water. There's also active transport. If some substance is going to cross a membrane, but it's going up or against its concentration gradient, energy must be put in. And that energy, typically in the form of ATP, again, that's the form of energy our cells use. And I've listed here some different types of um, active transport. One is pumps. I'll show you a picture in a minute. And then we have vesicular transport. Here's an example of a pump, and this is all of our cell membranes have this type of pump. The concentration of sodium on one side of the cell membrane is going to be greater than the other. So the concentration outside the cell, or extracellular fluid, that concentration of sodium is greater than what's inside. The sodium-potassium pump is continually pumping sodium out of the cell. At the same time, it's pumping potassium into the cell, also against its concentration gradient. That requires energy. ATP comes along, it gets split to form ADP, energy gets released to operate the pump. Again, active transport may be in the form of a pump. Endocytosis. This is where things are being brought into the cell, but the cell membrane is going to be involved here. And when you do any type of endocytosis or exocytosis, it's going to require energy. Phagocytosis, literally cellular eating. So the membrane comes up around this large particle and brings it into the cell. That is phagocytosis. Penocytosis, this is a cellular drinking. And this is where there's an invagination of the cell membrane that we see here. A bunch of fluid is going to come into the cell, and whatever solute is in that fluid also comes in. That's penocytosis. Receptor-mediated endocytosis. Well, some things may come along and we have something depicted as a star here, and it binds to a receptor on a cell, and that triggers an endocytosis, and whatever that is brought into a cell. Now, exocytosis is release of something from a cell. So the cell has made something and it's packaged it into what's called a vesicle. The vesicle is going to fuse with the cell membrane, and that substance, whatever it was, is going to get released. That's called exocytosis. Things like neurotransmitters, for instance, serotonin, it's a neurotransmitter. It will get released by exocytosis. But remember, any form of endocytosis or exocytosis requires energy. It's a form of active transport. Let's do a quick review of the organelles within a cell. Remember that there is cytosol, that's the fluid, and the fluid plus the organelles make up the cytoplasm. Some organelles, literally meaning tiny organs, some organelles will be bound by a membrane, have some sort of membrane around them, and some are not bound. The ones that are bound by a membrane, whatever happens within that organelle just needs to be contained. A lot of times there are enzymes maybe within the organelle that if those enzymes got out into the cytosol, it could damage the rest of the cell. We've already talked about mitochondria. You can produce lots and lots of ATP in the mitochondria as long as oxygen is present. Lysozymes contain these digestive enzymes. A cell always has some sort of cellular debris in it, old organelles that need to be digested, and that occurs in a lysosome. The rough endoplasmic reticulum, sometimes we call it the rough ER, and the Golgi apparatus, these are involved in processing and packaging of the proteins. For instance, in the previous slide, I showed you a vesicle with a substance in it. Well, that vesicle would have been formed by the Golgi apparatus. The smooth endoplasmic reticulum has to do with lipids that are produced within the cell. That's what occurs on the smooth endoplasmic reticulum. It also is very important for detoxifying. So we have all sorts of toxins that we eat, we drink, we produce, and the smooth ER will detoxify or neutralize those. Peroxisomes are involved with lipid metabolism and also can do some chemical detoxification. Non-membrane bound organelles. Ribosomes. Ribosomes are where proteins get produced, and we'll go through protein production in a minute. Nucleolus. This is within the nucleus, and it produces the ribosomes. Cytoskeleton has all sorts of proteins involved with it, gives the cell structure, makes it holds its form, helps with moving cell um, content from one place to another. The centrosome, involved with cell division. We'll talk about mitosis and meiosis in a minute. And then there are also proteasomes, and this can break down proteins. Sometimes a cell makes a protein, and it's not a normal protein, it's abnormal, and it needs to be broken down. Here's what some of these structures look like. You should remember these from basic biology class. Here's the mitochondria, lots and lots of ATP made within the mitochondria. The centrosome involved with cell division. Lysosome contains lots of digestive enzymes. The smooth ER, lipid metabolism, detoxifying toxins. Um, Peroxisome also has to do with some lipid metabolism. The Golgi um, apparatus up here, along with the rough endoplasmic reticulum, will package up proteins for secretion. Here's a Golgi vesicle. So here's a vesicle that can fuse with the plasma membrane. The nucleolus is within the nucleus. It's where ribosomes are made. Here are the ribosomes. This is the site of protein production. The cell membrane can have some modifications on it. Microvilli, cilia, and flagella. Flagella is a plural. A flagellum would be the singular. The microvilli, these are like finger-like extensions which increase surface area for absorption. Think about your small intestine. Your small intestine is where most of your nutrients get absorbed. And if you have more surface area for absorption, that's better. Cilia is present in the respiratory tract, plus in parts of the genital tract, and it's just these little extensions that help to move things along. And then the flagellum. And here we have the examples of that. Here's microvilli, increasing surface area. The more surface area you have, the more chances for things to get absorbed across. Cilia are larger, respiratory tract. Think about um, if you were to inhale something, some foreign particle, it can get trapped in the mucus, and then the cilia can sweep it up until you can cough it out. And then here is a flagellum on a sperm. Cells also are connected to one another. Some have what we call tight junctions, and here's an example of it on the bottom left here. All sorts of connections between the two cells. I refer to it as it's kind of like glue. It really keeps things from going between the cells. Desmosomes are more like rivets, so it's kind of a circular in one place and then another spot. Very strong connections. Gap junctions are these little tunnels. So if you see here, these little tunnels here, can allow ions to flow through. Cell division, mitosis and meiosis. Mitosis is when one cell divides into two identical daughter cells. Think about your skin cells. They always have to divide because you always have to create new skin cells. There are phases of mitosis. There's prophase, metaphase, anaphase, telophase. You can abbreviate it. Think of it PMAT. And then there's also an interphase. Now this is the time between mitotic divisions. During interphase, the cell is going to be growing a little bit. It's going to be producing new organelles, everything it needs to start to divide. It also is going to replicate DNA or copy its DNA. Here's an illustration of interphase and then mitosis. Starting with interphase, cells spend actually most of their time in interphase. And again, it's a growth period. It's time for the DNA, which is the chromosomes, to be replicated or copied. And then the cell will go into prophase, where the chromosomes become more distinct, and into metaphase. This particular illustration also adds another little phase in here, but we're going to skip over to metaphase, where the nuclear membrane has dissolved. We can see chromosomes lined up in the middle of a cell. Anaphase, the chromosomes are going to split, going into separate ends of the cell. And then telophase and and cytokinesis, the cell will eventually divide. Now the bottom line here, and we'll go into more detail in the semester, with mitosis, two identical cells are produced. Meiosis is going to give us 4 genetically distinct cells, and each of those cells is going to have half of the original genetic material, or DNA. Each of our cells contains 23 pairs of chromosomes, or 46 total. In meiosis, when the cell division occurs, the product is either going to be egg or sperm, and each of these cells will have 23 chromosomes instead of the total of 46. This is just a depiction of meiosis, and we'll go into more detail when we get to that chapter. First, there's an interphase. Okay, that's when the DNA is replicated. Your chromosomes are your DNA, and they have to be copied in order for any of this to occur. So here we have a pair of chromosomes. Okay, let's say it's chromosome number 4, and there's a pair of those, and each of those gets copied. So now you have what are called sister chromatids. Now the cell is going to go through meiosis. There's actually two phases of meiosis. In the first phase of meiosis, there's something called crossing over, where actually these chromosomes will swap genetic material. And that's what you see here, where this chromosome has a little bit of blue on it. This one has a little bit of red. Some chromosome material, or DNA, has been swapped. We call that crossing over. And then those two cells will go through the second phase of meiosis, meiosis 2. In the end, you get 4 daughter cells, genetically distinct. 4 daughter cells, genetically distinct with half the amount of DNA. So you started with this, look at those chromosomes, you have 2 total chromosomes, and you ended with these cells. So obviously there's half the number of chromosomes, but you can also see that each of these cells is different from the other cell, genetically distinct. All right, quickly, protein synthesis. Protein synthesis starts in the nucleus, and the nucleus here is depicted by the purple thing. This is your DNA, double-stranded DNA, a double helix. In a process called transcription, DNA is going to be read, and it's going to be transcribed into what we call messenger RNA, mRNA, and it's single-stranded. That messenger RNA leaves the nucleus, goes to the cytoplasm, okay, and that's where a process called translation occurs. And that's the sequence on that messenger RNA, specifically the sequence of those nitrogenous bases, is going to be read, and it's going to be translated into a protein. And again, proteins are made on ribosomes. And don't worry, we'll go into much more detail when we get to that chapter. And that does it for the module on cells.